What's a nervous system? Controller, communicator. To who? Well, the brain's part of the nervous system, but who's the brain talking to? Yeah, the rest of the body. It controls and communicates with different systems. And different systems like who? Yes, the muscular system, exactly. Why is it that my arm was just doing that? The nervous system told it to. I had conscious control of my nervous system, and I told it to. I said in my brain that I wanted to move my arm. We have conscious and unconscious control of different muscles in our body. What's some of the muscles we have control of through the nervous system, but it's unconscious? Your heart. I don't have to, thank goodness, because there's so many things on my plate, tell my heart to constantly beat. Do I? Whew, forget it, I would have been dead a long time ago. What else? Your digestive system. Well, so what do I need muscles for a digestive system for? Yeah, push things through tubes. Those are muscles that push things through tubes. And who controls those muscles? The nervous system. So, nervous system has a lot of control over what's going on in the body. Not only does it control things by using the cells of the nervous system, but it controls things by causing other organs to secrete chemicals that are then going to control things in our body. Is anybody who knows what that system is? Chemicals like hormones. The endocrine system, right? Aldosterone is an example of a hormone that's going to control something, change what a cell is doing. So the nervous system also has control over many of the players in the endocrine system, which are also going to control cellular function. All of this is done, fine-tuned, to help maintain what? Homeostasis. Now, I have to maintain a whole bunch of different levels in the body. As I'm standing here, I am working extremely hard. I am breathing so that I can regulate my respiratory gases. I ate breakfast this morning, so my digestive system's moving my delicious green shake through my digestive tubes. I'm making urine as I filter my blood. I'm also, in order to stand up here and talk to you, controlling tons of different muscles so that I can stand up here and I can talk to you. Since I'm talking to you, I have to actually form words and sentences, so my brain is working very hard to make sure that those words and sentences make sense. I'm pretty busy right now. Yes? And who's controlling all of that? The nervous system. Well, it's controlling it in two ways. It's causing things to happen, and it's also getting information from the world around me. Now, if none of you were here, would I be talking to you right now? And how would I know that none of you were here? That happened. You know what? I've been teaching for a long, long time. You already know, 17 years, right? Classes start at certain times all the time. I've been here since 2005. Yeah? 12:30 class starts, right? I go to my class, rush to my class, 12 o'clock. Swear that nobody's here. Where are these students? They have a test this week. How dare none of them show up? They probably all planned not to show up. There was nobody in my class because I thought it started at 12 o'clock. Hello? And then they started walking in, kind of looking strange at me, like, what's she doing? Because I figured I'm going to give the lecture anyway, because I'm not going to skip the material just because none of them showed up. Okay? Yeah, I'm losing it. Totally. So we get information from the environment that we're in. In that class, I got information that nobody was in the class because where did I get that information from? Did I go like feel all the chairs in the classroom? Where did I get that information from? I looked out into the classroom and there was no one there. I look out into this classroom and I see all these happy faces. What kind of information is that? It's sensory information. So information from the environment is sensory information. And it's going to help control the actions that I have to take in order to maintain homeostasis. So if I have to walk over there and pick something up, I know where to go, and then I have output information that's going to get me there. So when we talk about the nervous system, we have two basic divisions. We have information coming in and information going out. Sensory information is coming in, motor information is going out to cause an effect of something or another. In between, we kind of hand off the baton in the nervous system to different players in the nervous system. That's called integration. So if you look at the beginning of this chapter, you see the nervous system has three overlapping functions. Sensory, that's input. Integration, that's hand off to the next guy. And output is motor, an effect of some kind or another. Well, there's two divisions of the nervous system. There's what we call the central nervous system. That's kind of in the, why do we think we call it central? Anybody? It's in the middle, yes? Brain and spinal cord. And then we have everything feeding in and out of the central nervous system. What do we call that? It's on the outside. Peripheral nervous system. So these next 4 chapters are going to talk about how these cells work, and that's pretty much this chapter, fundamentals of the nervous system and nervous tissues. And then how all these players play their role in controlling body function. So you're going to see this diagram about a bazillion times in the next 4 chapters in some way, shape, or form. But this is the basis for the nervous system. So we start up at the top and we see the central nervous system. And what's the central nervous system consist of? Brain and spinal cord. And then we have what we call the peripheral nervous system. Another thing I want you to notice, the arrows connecting all of these different parts in the nervous system. So does information go in and go out of the central nervous system to the peripheral nervous system? How do you know that? Hey, you get 2 arrows, some going in, some going out. You see the bottom of the diagram, what it says? The peripheral nervous system, we have things going out to where? And notice they just go out. They don't go in. Things like muscle contractions, automatic things, thought about things, things that speed things up, things that slow things down. The things I'm talking about are effects. And then I also have information coming in through the peripheral nervous system. We call that sensory or afferent. So motor is efferent, exit, out, and sensory is afferent, coming in. Some of our nervous system is going to control body functions. Somatic. Somatic motor is voluntary. The thing I just did with my arm. Yes? Was that voluntary? Yeah, sometimes it happens and I don't even think about it. As a matter of fact, most of the time, because we've done it so many times, we don't think about it. It's still voluntary though, because we can make it happen and we can make it stop. Skeletal muscle is usually what we're talking about there. Now, what was skeletal muscle? When we talked about tissues, skeletal muscle is what? It was striated, remember that? And we can cause the cells to shorten and lengthen. That's the job of the somatic nerves, the motor division of the peripheral nervous system, the somatic nervous system. Then we have stuff called autonomic. And sometimes visceral things are autonomic. What do you think autonomic is? Yeah, kind of automatic. They're controlled by the nervous system, but we don't have to constantly think about them. So we call them involuntary. Nerve impulses to heart, smooth muscles in the digestive system, or big muscle that helps me breathe. What's that called? Diaphragm. Your autonomic nervous system is always going to take over. So even if you try to stop your breathing, you can't do it for too long because your autonomic nervous system won't let you. So the autonomic nervous system and the somatic nervous system play a role in breathing, mostly this guy. We can speed things up, we can slow things down, and we'll talk about the parasympathetic and sympathetic divisions of the autonomic nervous system. You see how my words are starting to get really big? But understand this and you'll understand what I'm saying, okay? Because they're all tied in to either the peripheral nervous system or the central nervous system. So understand this diagram, because this is kind of the summary diagram for the next 4 chapters. When you look under the microscope, and you'll probably see these next week, you probably won't get to them this week in lab, but when you look at them under the microscope, cells of the nervous system, and what are those cells called, by the way? Does anybody know? They're called neurons. Are gigantic. Now most of the cells that we look at under the microscope, when we take our little slide and stick them under there, we can't really see them, can we? Until we magnify them under the microscope. But when you pick up the slide for the neurons, you can actually see them before you even stick them under there. They're about as big, at least the cell bodies of those cells are about as big as the letter E you looked at in the beginning. They're huge. In the cellular world, that's huge. They're so huge that they need help. They need help taking care of themselves. The other thing we're going to find out about neurons is once I have them, that's it. So in your nervous system, for the most part, all of the neurons that you'll ever have in your lifetime, you've got them already. You had them when you were a baby. If you kill them, what are you doing? You going to get any more? Nope. What kind of things can kill them? Injury? Toxins? Toxins like what? Alcohol? Drugs? Can kill your neurons. You don't get any more. That's it. There are some exceptions to the rule, and we'll talk about them second semester when we start talking about the special senses. So the little receptors on the surface of your tongue. Thank goodness. You ever go to the pizza parlor and get a nice big hot slice of pizza? You can't wait. Got to eat it right away, and you burn the living daylights out of your tongue. And then you'll never taste again, right? Yes, you will. because that's one of the exceptions to the rule. So you can remake more neurons. Did you know that neurons controlled your taste? Yeah. What else? Smell. Those are neurons. They're going to send information to your big fat file cabinet. That's what I call this guy up here. Part of your central nervous system, your brain. You have tons and tons of files, information that you store in your file cabinet. And every time you go and interact in your environment, you're going to pull information from your file cabinet. Did you have all those files when you were born? No. You had the space, but you didn't fill up the file cabinets. So think of the spaces in your brain, different spaces in your brain, as big empty file cabinets. Tons and tons and tons of them. And as you get older and older, you're going to collect information and store them in different file cabinets in your brain. Different groups of cells. So when I stand up here and I look at this, I see this is green and it says Celtics on it. God help us, Lord knows what our season is going to be this year, but once a fan, always a fan. Yes? Where did I learn that? Did I know that when I was born, that this was green and it was a lanyard and it said Celtics on it? Nope. That file cabinet was empty. What did I have to do? Learn. I had to learn. And when you learn, you put a folder in your file cabinet. And we're talking about everything. You learn everything. Some things you have pre-set up files for, just like when you buy a new computer, you get, some standard files. You have some standard files when you're born. But most of the stuff you have to learn. I have to learn that's green. I have to learn what the touch of this feels like to understand what it is. You learn everything. And that's what you're doing, is relying on the files that you've created when you learn something in the nervous system to understand what this is, to read language, to understand speech, to move. Did you know how to do all the movements you do now when you were first born? Guys, you couldn't even hold your head up, right? So what did you have to do? You had to learn. You had to learn how to walk. You had to learn how to ride a bike. You had to learn how to drive a car. It feels like you do it automatically now. But think about when you didn't know how to do it. Yes? You had to make files for that. And that's what you're constantly going back and relying on. So these cells are constantly communicating back and forth, grabbing some information for your files and acting on that in the peripheral nervous system. In order for these cells to do what they need to do properly, they need help because they're so big. And that's where we talk about a group of much, much smaller cells. And we see most of them in the central nervous system. We see a few of them in the peripheral nervous system. They're supporting cells to the nervous system. The neurons, the business end of the nervous system. They call these guys neuroglia, or glial cells. So there's different types of neuroglia. We're going to find them in different places, and they have different jobs. The first guy here is called an astrocyte. And an astrocyte is most abundant in the central nervous system. It's one of the most abundant of the neuroglia. And it is going to help hold things in place for neurons. It's going to help connect the neurons to things. Like, for example, in this diagram, what is the astrocyte connecting the neuron to? Capillaries. Well, what do you think it's doing that for? What does the neuron need from capillaries? Nutrients. Have to get some nutrients. Nutrients to do what? Yeah, to stay alive, to do the work it needs to do. It's got to do things. It's got to make things. It's got to carry on day-to-day function, and it needs nutrients from the capillary beds to do that. When it carries on its day-to-day functions, it also generates what? Waste. Well, how's it going to get rid of waste? Through the connection through the astrocyte to the capillary beds. So when we talk about the circulatory system in second semester, we're going to talk about the fact that those capillary beds are going to bring nutrients to the cells and remove waste products for the cells. So astrocytes are sort of that connection. They provide support and connection. The next group of cells are called microglia. And these little guys are in the central nervous system still. They have these little thorny processes. We're going to talk about processes in neurons. And what's a process in a neuron? Something that sticks out. Yes? So what do these little microglia cells do for a living? Did anybody read this chapter? You did? Pipe up. Feel free. They're macrophages. Well, like macrophages. What's a macrophage? You learned that. They get rid of debris. Any debris in your central nervous system? Probably. Do we want it just building up and hanging around? No. It'd be nice if we could have somebody who could clean up. That's what our microglia are going to do for us. The other thing we're going to find in the central nervous system is fluid. And one of the ways that all the cells get nutrients that they need is through the fluids that surround them. These microglia called ependymal cells. Ependymal. What does that kind of sound like? Something you already know. Epithelial, yes? Didn't epithelial tissue kind of all line up together nice and tight like little soldiers? Is that what those guys are doing? These guys are going to help produce those fluids that are going to help the central nervous system function correctly. We're going to find it in spaces in the brain. Yes, you have spaces in your brain. Big old spaces, cavernous blank spaces. Yes? Do you know what they're called? You're going to hear this word again when we talk about the circulatory system. They're called ventricles. And you have 4 major brain ventricles that are filled with fluid. So these guys, the ependymal cells, are going to make that fluid. Now they are very precise on how they make their fluid. Anybody know what that fluid is called? Cerebrospinal fluid. Guess where it is? The brain and the spinal cord. So these guys are responsible for making that cerebrospinal fluid, which sometimes we refer to as the ultrafiltrate of blood. Very particular what I'm going to put in my fluid that's going to surround my central nervous system. I just don't want to let anybody in there. These guys also create what we call the blood-brain barrier. So if I'm a drug developer and I want to make a drug that affects a certain portion of your brain, what do I have to do in order for that drug to work? I have to get it by these guys. Sometimes developers will work for years, find the most amazing drug that will do the most amazing thing, but then when they actually give it to you, those cells say, nope, not letting that into my spinal fluid. Sorry. Back to the drawing board. Okay? So that creates an ultrafiltrate of blood plasma. It's not going to let just anything in. Unfortunately, sometimes it lets nasty things in too, but that's what it does for a living. Another group of cells we're going to find still in the central nervous system. So all these guys are still in the central nervous system. are groups of cells that are going to act as insulators. They insulate some of the projections in our neurons. And they insulate them so electricity doesn't leak out. What's she talking about, electricity? When we talk about a nerve impulse, we're going to talk about electricity. The movement of charged particles across a membrane. That's what causes these cells to do their thing. In a few moments, we're going to start talking about something called an action potential. And the chemical change that goes on in these cells in order for them to do their work. Wires in your house, are they just all kind of hanging out? No coverings on them, all touching each other, just, yeah. If they are, that's not good. Fix that. Do you ever go down in the basement, look at the wires in your house? What are they all covered with? Just some little plastic stuff, right? Different colors for different ones sometimes. So they can let the electrician know who does what for a living. Yes? But what are those coverings for? Yeah, they insulate, so the electrical current that's passing through those wires doesn't do what? Transfer to their neighbor or transfer to the wood that they're around. Would that be a good thing? No. That would cause all sorts of problems, wouldn't it? Anybody ever been electrocuted? It's not pleasant. So we need to insulate those wires in our house. We also need to insulate the wires in our nervous system. So the insulating cells, and they act kind of like, do you ever take a tube of toothpaste? And this is old days. You know, nowadays you get all these fancy little contraptions. But you take a tube of toothpaste and you get one of those little, looks like a little key thing on the end. You stick it on the end of the tube, and I'm wicked cheap, so I'm getting every ounce of toothpaste from my tube. I don't know about you. But you wrap the tube around that little key thing until you squeeze all the stuff out of the tube. Well, these cells sort of do that, act like the tube around the little key thing. They wrap themselves around the projections of different neurons. These cells are called oligodendrocytes. And we are going to find them. They're the kind of the gray thing that's wrapped around some of the nerve fibers that help to insulate them. They create a sheath of, it's like lipid-like material. Because most of the cells, at least the surrounding part of a cell, is what? That membrane that surrounds the outside of the cell. What's most of it made of? Phospholipids. So it's a lipid-like material, phospholipid, because most of the cells squeezes itself to that, and it's called the myelin sheath. So in the central nervous system, we find oligodendrocytes producing the myelin sheath. These guys produce the myelin sheath in the peripheral nervous system. So now, finally, we're talking about some glial cells in the peripheral nervous system. These cells create the myelin sheath or the insulating sheath in the peripheral nervous system. They're called Schwann cells. And they do the same thing. They kind of wrap around the outside of the fibers that are leaving the neurons. We'll talk about those guys in a minute. We also find little cells called satellite cells, also in the peripheral nervous system, and they're usually associated with the cell bodies of neurons. And remember, neurons are huge. I'll give you an example. I have a group of neurons whose cell bodies are up here in my lumbar region, and whose fibers, and this one happens to be the axon, go all the way down my leg. That's one cell. We have groups of them called nerves. So when I talk about one cell, I refer to it as a neuron. When I talk about a group of them, just like wires in your house, I'm going to call them nerves. You with me? All right. So satellite cells, Schwann cells, we find them in the peripheral nervous system. Astrocytes, microglia, what's the third one? Ependymal cells and oligodendrocytes are found in the central nervous system. All right, so this is the neuron. By the way, where are you going to see that again? On your third lab exam. So you should know the parts of a neuron. Remember we had cells, we went through all the parts, endoplasmic reticulum, ribosomes, all of that good stuff. Well, this is a cell just like any other cell, except this cell is huge and has these big, long projections. The cell body contains what? It's that big round hunkin' organelle, the only one we can see under the microscope. Nucleus. So the nucleus is contained in the cell body of an axon. These cells last, or can last, an entire lifetime. So they have a lot of longevity. They don't have, for the most part, the ability to divide. Because they're so big, they use a lot of energy. They have a very high metabolic rate, the rate at which their chemistry takes place. So what's that mean? How do I make energy? Do you know how I make energy? Of course, this is second semester when we talk about cellular respiration. But what do I use for energy in the body? ATP. ATP molecules. Those are my energy molecules. They're kind of like the batteries that drive my cells. And what molecules do I use to make ATP? Carbohydrates. Simple ones. Like, you remember what a simple carbohydrate was? What was the most popular one? What kind of sugar? Glucose. So does my nervous system need glucose on a regular basis? Yes. Does it need glucose when I'm sleeping? Yes. Because I'm not just sleeping when I'm sleeping, am I? No. No, 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 no. I'm sending messages out to the heart and to the respiratory system. I'm sending messages out to different muscle systems. I'm doing a whole bunch of work when I'm sleeping. So I constantly need glucose. Who didn't eat breakfast this morning? Who's very mad at you right now? Your neurons. You're not getting enough glucose. You go on that, I'm going to be beautiful in a week diet. No carbohydrates. Yes? Do you like that one? You're supposed to get about 200 to 300 grams of carbohydrates a day on a regular basis. And some of these diets tell you, oh, no, no, carbohydrates are evil. Has anybody ever been on one of those wonderful diets? Yeah. What do they tell you can have?
They want like under 30.
Yeah, like 20 grams.
Yeah.
Okay, so what's the problem with that? It's evil. Why is it evil?
It's not enough.
For who? Your brain, which is kind of important, yes? Yeah, kind of important. And guess what? Your brain, your neurons, are only going to accept one form of molecule to make its energy. And you know what it is? Glucose. You can use other things. Your muscles aren't too picky. They're happy with fats and stuff like that. But your brain says glucose, and only glucose. Very exclusive. So glucose is extremely important because of its high metabolic rate to make sure I get enough of it so it can do its work. So the neuron cell body is where we're going to find the nucleus, where we're going to find all of its genetic material, We're also going to find little chromatophilic substances or Nissl bodies, and they stain up as little dots. Tons and tons of mitochondria are going to be scattered throughout this cell. Why is there going to be tons and tons of mitochondria? Because I got to make a lot of ATP. So we're going to find them scattered throughout the cell. We're also going to find extensions, things that stick out of the cell, things that stick out at the top and then feed out at the bottom. We have neuron processes. We have some that sort of feed into the cell body. What are those guys called? Dendrites. They're the receiving end of information. They're going to help bring information into the cell body. It's kind of like the peripheral nervous system and the central nervous system. If I look at a neuron, the central part, that's the what? Central part of the cell. What is it? The cell body. And the things feeding in and feeding out are dendrites, and the other group of processes that feed out are axons. Now, feeding in and feeding out, what in God's name is she talking about? Feeding what in and feeding what out? Remember I talked about that electricity stuff? That's what these guys are accepting and transmitting. So changes in or movement of charged particles through this cell is how it works. So those processes, those little fibers, are where we're going to accept information in the form of chemical change and send information out in the form of chemical change as well. So axons feed information out, dendrites information in, and then what's all that little stuff around the axon called? The Schwann cells in this one. So what kind of nerve must this be? Is it in the central nervous system or is it in the peripheral nervous system? It's in the peripheral nervous system. How'd you know that? Because there's Schwann cells there. What would they be if it was in the central nervous system? Oligodendrocytes. Okay, so Schwann cells, I know it's peripheral nerve. That's going to act as my insulator. Insulator so I can keep that message going without it leaking out the sides. Just like the wires in your house. You don't want the wires in your house to fire off wires beside it. Do you want wires in your nervous system, axons in your nervous system, to fire off random neurons willy-nilly? What's that called? That's called a seizure. Is that good? No. It causes all sorts of problems, sending messages to places where we don't want to send messages at any given time. So the myelin sheath is extremely important. The other thing we see is little spaces in the peripheral neurons between the myelin sheath. Those are called the nodes of Ranvier. And sometimes when a nerve impulse is described, people describe the nodes of Ranvier as a place where electrical message jumps from one place to another. Well, that's not what happens. Nothing jumps out. We don't want anything to jump out, do we? We want to keep it moving through the fibers. So message actually gets pushed through a little bit faster through those little spaces or nodes till it gets to the next portion of the fiber that's leaving the cell. So, different neurons look different. This diagram on page 392 is a diagram that shows us the myelin sheath. So when I look at this and I make a cross-section of a neuron, the axon is in the center there and the myelin sheath is around the outside. Again, remember, that's an insulator. All right, different types of neurons. information coming in, information going out in different ways. So the next part of the book talks about some of the different structural classifications of some of the different neurons we're going to meet in this following 4 chapters. We have something called a multipolar neuron. And what does a multipolar neuron look like? Well, multi means what? Yeah, more than one. So they have 3 or more processes. Typically one axon coming out the end and a whole bunch of dendrites feeding into the cell body. These happen to be the most common type of neurons in humans, with more than 99% of the neurons belonging to this class. That's why he gets top billing when we show you a picture of a neuron, and we want you to know the parts. Then we have something called a bipolar neuron, a little bit different. Big long dendrite feeding in, big long axon coming out. We're going to talk about these cells when we talk about some of our senses, like our sense of sight. You'll see that term come again in 2nd semester. And then we have something called a unipolar neuron. Weird little shape associated with the central nervous system. And we don't see as many of those guys around. So, we see some of the differences between the different types of neurons. General definition for that, that's all I want you to know. Sensory neurons versus motor neurons. Sensory are bringing information where? Into the central nervous system. Motor information coming out of the central nervous system to cause an effect. So we see on this part of the diagram some of the different places we find these multipolar, bipolar, and unipolar neurons, bringing information in, bringing information out of the central nervous system. When you feel something, why is it you feel things? And sometimes you can stop feeling things. When you stop doing what? Sending message. Don't ever talk to your mother on the phone while you have a big sharp knife and you're making dinner, because you can cut things. As I did one day when I took my plastic bag of meat and I couldn't find my scissors, and I was on the phone. You get catching the picture? And I was holding the bag, and I had my nice sharp knife that my son was kind enough to have just sharpened for me. Thank you, son. And opened my meat bag. Well, this finger was in the way. And I thought I just got a little cut, so I was like, okay, I just cut my finger, I'll call you back. Didn't even hurt. And then it was bleeding pretty good. So I looked in the sink, and the pad of my finger was in the sink. I said, oh goodness, that's probably not going to stop. Let's go to the emergency room. It was a full moon that day, so I got to sit in the emergency room that evening for quite some time. You know, anybody who's worked in the hospital, it's the truth. Full moon, you're not getting a break. No, 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 no. I can no longer feel anything, because what did I do? I mean, it's still there. If I push hard enough, it actually hurts. But I no longer have that fine sense of touch on this finger anymore. Because what did I do? Those receptors for that are gone. I cut them off. Now what's there? Scar tissue. Can I feel anything with scar tissue? You want to be really tough? Little kids love this. I can't roll up my sleeve, but is this part of your elbow? Grab the skin and pinch as hard as you possibly can. Go ahead, do it. Really hard. Any pain? No. And you go, I'm tough. Pinch that as hard as you can. And they'll pinch it and they're like, nothing. You're going to get a big bruise because you're damaging cells afterwards, just to let you know. But you don't feel any pain. Why? Because there's no what there? There's no receptors for pain. You getting my point? All of these cells, their communications, if they're disrupted or they're removed, you're going to stop feeling those things or processing those things when we break up some file cabinets in our head that are responsible for filing certain things. When we discuss the nervous system second chapter, and we actually talk about the brain and where certain things are kept, we're going to see if we damage certain portions of our brain, we're going to have a hard time processing information. So pain, sensory information, receptors that are going to send information in. Now what's this information I keep talking about? And that's what the book is going to talk about next, when it talks about something called membrane potentials. So we're going to go back to the chemistry that scared the crap out of you in chapter 2. We're going to talk about it again when we talk about nerve impulses. Remember when we talked about osmosis and diffusion and how molecules tend to go with their concentration gradient? What does that mean? They like to even out. They go from a high concentration to a low concentration, yes? Well, when we talk about membranes, plasma membranes, they are a little different because they have these little channels in there that are going to help regulate ion movement across them. These guys create something called membrane potential. And I think I mentioned it way back when, but maybe I didn't. What's the charge associated with a plasma membrane? A charge caused by what? Again, visit your chemistry brain. Dust off the chemistry brain. Charged particles are what? Ions. When I lose an electron, I become a what? Positive ion. When I gain an electron, I become a negative ion. We have these floating around all over the place within our cells, on the outside and the inside. And these little proteins are going to help regulate their movement. I have one little gate, and we're going to call them gates because that's kind of what they're like. They're going to let things in, let things out. Remember, we're talking about a semi-permeable membrane here, right? I'm visiting all the information you already know, yes? So this semi-permeable membrane has these little ions floating around it at all times in the fluids that surround it. And these little proteins are actually going to work against the concentration gradient when a cell is at rest. So when I have a neuron that is at rest, resting membrane potential is associated with a certain charge around the cell membrane. It's more negative on the inside and it's more positive on the outside. And the reason it's more positive on the outside is because of these little proteins that actually push ions against their concentration gradient. What would that mean? If I push you against your concentration gradient, I'm pushing you from a low concentration to a higher concentration. Well, what the heck am I doing that for? That's the way the cells were made, but it kind of comes in handy when we ask our nervous system to do something. So resting membrane potential is typically associated with a charge that's more positive on the outside and more negative on the inside. When I have something that is two opposite sides, kind of like when I talked about covalent bonds that didn't share fair, what kind of bond was that? Two opposite sides. Two opposite sides of your Earth. Separate poles. Yes? So if I have two opposite charges on my membrane is polar or polarized. That's going to be important when we talk about the change. When the change comes, the flow of charged particles across a membrane, the electricity part of this whole deal, we're going to see that the membrane will become depolarized. That's another name for neurons firing off. We also call it an action potential. Did I lose you yet? All right. So these guys, these ion channels, are responsible for regulating ion movement across a membrane. Now, these channels can be affected by two major things. One, they can be affected by chemicals. In this one over here, we see a chemically gated ion channel. And in this case, what's the chemical that comes in contact with it to cause it to change what it's doing? Sodium. See those little yellow balls? These guys can also be regulated by other chemicals, hormones, other chemicals that neurons give off to pass their message to the next guy in line. You know what those chemicals are called? Neurotransmitters. So when we talk about nerve impulses, we're talking about changes in some of these ion channels, these protein pumps, caused by different things like changes in ion concentration or chemicals like neurotransmitters or chemicals like hormones. You with me? Other things, too much sodium, too much potassium, too much chlorine, all charged particles, can also affect some other channels. We call these channels voltage-gated channels. So a change in charge or a change in chemistry, chemicals, can affect these ion pumps in different ways. You want to kill somebody and not leave a trace? What could you do? No, that would be too radical. What if I gave you like mega doses of potassium into your circulatory system? Really, really quick. Do you know what's going to happen? Your nervous system's going to totally freak out. Especially the portion of the nervous system that controls your heart. Yes? Just from a simple ion. So, ion, voltage, and chemical gated channels are what help to control the nervous system. And here's how the nervous system works. So the next thing they talk a little bit about current and resistance, and they do the whole little physics things there. I'm not going to get crazy about that, so don't worry about it. Ohm's law talks about how fast things move across the membrane. I'm not going to make you memorize that stuff. But you should know the difference between the different types of channels and what affects them. Resting membrane potential then, positive on the outside, negative on the inside, is what our neurons kind of hang out as until they have work to do, pass a message. They talk about some of the differences in plasma membrane permeability. Now typically all these ions would even themselves out on either side if it wasn't for those little protein pumps. But what we're going to see is we're going to see a much higher concentration of sodium ions on the outside of a cell versus on the inside of a cell. And this is going to play a big role in nerve impulses. If I can disrupt this balance, if I can allow sodium to rush into the cell, what's going to happen to the charge associated with the cell? If sodium, tons of sodium rushes into that cell that's just sitting there right now, what's going to happen to the charge? It's going to become more positive. So if I had a little voltmeter, and that's what that is, to check and see what the charge was associated with a cell at rest, resting membrane potential, I would see that it would be measured in millivolts at about minus 70 millivolts when the cell is at rest. And who's going to be responsible for keeping that charge the way it is? The little pump called the sodium-potassium pump. So if you look at the focus on page 398 in your textbook, they talk to us about the sodium-potassium pump. And the sodium-potassium pump is the gatekeeper, the keeper of the charge associated with any given membrane. And it's going to do that by moving sodium-potassium against their concentration gradient. So if I look at the concentration of potassium inside the cell versus outside the cell, I'm going to see it's much higher inside than outside. If I look at the concentration of sodium inside versus outside, I'm going to see it's much higher on the outside than it is on the inside. And again, that is during rest. The other thing I want to point out before I forget, you have a program that you probably want to crack open now. The program is called Interactive Physiology. It came with your textbook. Did you guys get an actual physical disc? All right, if you didn't get a disc, don't panic, because you know where you can find it? Mastering. So when you log into Mastering and you go into the study area, has everybody done that? Those of you who don't like your test grades, you probably should do that. Yes? You can go in there and find a whole bunch of stuff. But you can find in there on the left-hand side a little tab that says Interactive Physiology. And in Interactive Physiology, there's different modules, different groups of information for different topics. And the first one you're going to start to crack open is this one, the nervous system. So if you go to Interactive Physiology, you can start looking at the function of a neuron. But because I look at neurons and I look at neuronal changes, what I'm actually looking at is changes in charge, changes in electrical current that are being passed. And I can actually measure that. I can pick up those electrical changes on these tracings. When a membrane is at rest, again, minus 70 millivolts is where we're at. In this diagram, we see depolarization and hyperpolarization. Now, if I drew a line and went up and down around that minus 70, that's pretty much what's really going on. It's not constantly going to be minus 70 all the time, is it? Because I'm pushing things out, things are going to move back in. So we're constantly seeing a line like this that goes around that minus 70. When the line comes above minus 70, it's called depolarization. When the line goes below minus 70, it's called hyperpolarization. So we see different grades of change within cells, and that's what they talk about under graded potentials. In order for the cell to pass its electrical message, it has to reach a certain point. A change has to reach a certain point at a certain place in the cell. This is constantly happening. So if it was that sensitive, what would constantly be happening? I'd constantly be firing off all my neurons. So these simple changes aren't going to cause a total cell change. A total cell change would be a true action potential, where the entire axon of a cell would depolarize and then repolarize when the neuron was at rest. So, what causes depolarization and repolarization of the whole cell? And that's what they talk about under action potential. So what we're going to see is a big change in charge. That's what's going to cause a big change in my cells. That's what's going to cause a wave of change down the axons of my cells. So remember sodium? Sodium was the guy that was going to help me with the change, right? And those little pumps were the guys that were responsible for sodium. Yes? So when we talk about a nerve impulse, we talk about something called an action potential. And the role of those little pumps and the concentration of sodium inside the cell. When everything is at rest, those gates are going to make sure everything stays around minus 70 millivolts. But what if something comes in contact with a whole bunch of those gates? And remember, information comes into a neuron via the what? The dendrites. So we have a bunch of these little sodium-potassium pumps all over the dendrites of any given neuron. And again, making sure that everything stays in place during resting state. But what if something came along and opened up a whole bunch of those gates and caused a lot of sodium to come in? What would happen to the charge inside the cell? And think of this as happening in a wave. So dendrites kind of sticking their fingers out here. All of a sudden, a bunch of little chemicals come in contact. Let's say neurotransmitter comes in contact with a bunch of those little pumps on the dendrites. It's going to cause them to open up, and sodium is going to flood into the cell. And it's going to come in a wave. That wave is then going to spread through the rest of the cell. But once that wave of change in charge hits a certain point, not just a little wave, it's going to be a good-sized wave, yes? It's going to cause change to happen throughout the whole cell. And that's called depolarization of an entire cell during an action potential. So what is that point? That point it has to reach, that amount of change it's got to reach to actually fire the whole cell off. Well, that point is called threshold. And in this diagram, we see threshold is what? Minus what? Can you see it in the diagram? Minus 55. Minus 55 millivolts. Once the wave comes in and it changes the charge just enough to hit minus 55 millivolts, that's when the business is going to happen. And it can't just happen on the ends of the cells. It's going to come in a good-sized wave and hit an area of the cell called the axon hillock. So I hate to go back, but I'm going to go back. When I look at the cell, I'm talking about this here. Dendrites, all those little pumps, cell body, and right here is the axon hillock, and it's labeled in the diagram. That wave has to come in and cause enough change here to then fire off the rest of the cell. What if little waves come in? Is anything going to happen? No. Happens all the time. Graded potential. But a big wave has to come in to cause depolarization of the whole cell. When I look at this on my pick up the electrical charge change, I see that I go up to about plus 30 millivolts because tons and tons of sodium is going to come into the cell. Once I hit that axon hillock, all the little receptors all the way down the axon are going to start to do what? All those little protein pumps are going to start to open up, and sodium from the outside is going to rush in and bring the charge at full depolarization up to about plus, oh, wait a minute, wrong way, plus 30 millivolts. Then what happens? What's happening to the charge as we start to go down the other end of the peak? What do you think might be happening to the cell? Whatever it is that caused those guys to open is eventually going to stop doing that. And what are those pumps going to then eventually start doing again? Doing the business of pumping out sodium, yes? So what's going to happen as they all start doing their job again? What's going to happen to the charge inside the cell? It's going to start coming down. That's called repolarization. We're going back to that polar membrane. Sometimes we go a little bit further down. What's that called? Hyperpolarization. And those are the different parts of an action potential. Those are what happens to the cell during a nerve impulse. So you should understand all the steps of the action potential. This little diagram down here is kind of neat because it shows you who's rushing in and who's rushing out. Sodium permeability of the cell during depolarization is going to be much, much greater. What the heck does that mean? Depolarization, increase in charge, sodium's rushing in. Sodium permeability is going to be what? What does permeability mean? The ability to come in, yes? So sodium permeability during depolarization is way, way up, yes? What's it going to do during repolarization? Start coming down, because we're going to start pumping it where? Start pumping it out of the cell. Okay? So I'm not going to say any more than that because I think I've just blown your brain up. I want you to get through action potential. I want you to understand it. I want you to go use interactive physiology because that goes over what I just talked about, the whole little action potential thing. The nice thing about it is it's got pictures. You can sit and listen to it and you don't have to read a book. Oh, so it's a great study tool. Make sure you pick up your tests. Make sure you sign in.